Welcome to the After On podcast. I'm your host, Rob Reed, and this is a series of conversations with thinkers, founders, and scientists. Take a little time and stretch out, because these talks are unhurried and meant to bring you to a top percentile understanding of something important. So, whether you're into startups or ideas, a techie or a lit major, take your time, engage your mind, and you'll be glad you did it. Especially this week, when we'll be talking to Chichinilsky. EJ is a professor of neurosurgery and ophthalmology at Stanford. He also co-leads Stanford's Neurotechnology Initiative. EJ and his team are creating a device that will literally bring sight to the blind. That device, of course, is fascinating, as is the science it's built upon. That science and the research that's underway in EJ's lab is on the outer frontier of what we know about the human brain. EJ and I are going to explore all of that in terms that non-experts and non-scientists can understand. If you're new to this show, welcome. It features in-depth conversations with world-class experts about their domains of knowledge. In each episode, I try to bring you, and myself, a serious grounding in something complex and important that we should all probably understand a bit better. If you enjoy this episode, please browse the show's archives at after-on.com or in your favorite podcasting app. You'll find in the archives all kinds of fascinating conversations with some of the top people in robotics, neuroscience, cryptocurrency, drones, augmented reality, synthetic biology, and many other fields. If you're interested in these sorts of things, I hope you'll consider subscribing to the show. Before we start, a quick note for the 950-ish people who support this show on Patreon. As I often do, I'm publishing an abridged version of today's episode to the main podcast feed, out of respect for the time of my more casual listeners. And you are now listening to the main podcast version, in other words, the shorter version. I'm meanwhile posting an extended version of the interview, the director's cut, if you will, to my Patreon feed, which is at patreon.com/RobReed. That long version is available to those who support the show with monthly contributions of $5 per month or more. And of course, that version is also seamlessly and portably available to my backers through their podcasting apps. So if you are a backer, it's probably on your phone right now. I'll talk more about Patreon in my comments after the end of the interview, but for now, I just want to urge my Patreon backers to stop this recording and go directly to that extended version. It's maybe 30% longer than this version, and EJ's work is fascinating and sophisticated enough that it's really worth hearing every syllable. To the rest of you, fear not. The version you're about to hear is also utterly intriguing. It is not dumbed down, and you're about to learn a ton about how vision works, how the brain works, how blindness may be cured. And then toward the end of the interview, EJ and I have a fascinating speculative conversation about where neural enhancements in general could be taking us, and how they could ultimately change what it means to be human, perhaps within our own lifetimes. And with that, let's join EJ. So EJ, thank you so much for making time in your very busy schedule to share some of the amazing scientific and engineering work that keeps you so extraordinarily busy.
It's my pleasure. I'm glad to be here with you.
Now, before we get into your own approach to the problem of blindness, which is quite unique in the emerging field of retinal prosthetics, I'd love to briefly talk about the current state of the art, the devices that are already available, how long they've been around, their capabilities and limitations, and so forth.
There are a couple of devices that are out there on the market that are implanted in the eyes of profoundly blind people and electrically stimulate the neurons in the retina in response to an image that's captured by a camera. Those devices cause people to see flashes of light of various forms. They are a proof of principle that one can actually restore some form of vision to people who've been blinded by photoreceptor diseases like retinitis pigmentosa and age-related macular degeneration. And they really show an extraordinary thing. You can take somebody who hasn't seen anything for 30 years and electrically stimulate their retina, and sure enough, they report seeing things.
Now, when you say flashes of light, I'm assuming that those flashes in some way correspond with the visual environment around the person, or they wouldn't be at all useful as a retinal prosthetic.
That's correct. The devices capture the image in a couple of different ways. There are two different major devices that are out there. And based on the image, they stimulate particular electrodes on the retina And those electrodes pass current, and the current that they pass causes neurons in the retina to be electrically activated. And then those neurons send visual information to the brain.
And what kind of seeing do the patients experience? Do they see faint, shadowy outlines? What level of sight is it that they're suddenly able to access?
It's quite variable, but I would say the dominant, interesting percepts that are elicited are irregular forms, blobs or streaks of light that are elicited by electrically stimulating the retina. And they're difficult to put together into any kind of image. They're large instead of being small and granular. And it's a little bit hard to know how they're going to relate in a sensible way to the image because the forms that the electrodes elicit are very coarse and irregular and poorly controlled. The vision that's imparted by these devices in human patients is really quite minimal. And I don't mean to be critical because these devices are the proof of principle that is opening up an incredible field. I have profound respect for the work that was done. On the other hand, patients would not give up their cane or their guide dog for these devices. The cane and the guide dog are far more useful in terms of daily living than these devices.
And those blobs and those coarse shapes that are seen do correspond to what's in the user's environment. So I'd imagine, particularly perhaps in an environment that they're familiar with, they might spend an enormous amount of time in 3 rooms in their house or whatever it is. The correlation between blobs and aha, that's the dresser, maybe that gets a little bit more instinctive for them, whereas out navigating in the real world with novel sights and novel shapes and blobs always around would be a little bit tricky.
Yes, and another way to put it is when you frame the task for a person, you can make this device more useful. So for example, if you say, look, you need to step forward and there's a doorway and it's either a little bit to the right or it's a little bit to the left, and the doorway leads into a room with a bright light, and you want to use that information, you can tell that there's a bright light over on your left or over on your right with one of these devices. You can distinguish that. And so you could use that to have a slight improvement in your behavioral outcome by going in the correct direction. So if you dramatically simplify the task that's being performed, you can demonstrate some utility. On the other hand, normal vision, what we experience in our normal visual world, isn't like that. We're very rarely faced with extremely simplified, rudimentary choices that we use our visual system to make. We're typically doing things like recognizing complex objects, reading facial expressions, reading text, navigating a new environment or a changing environment with objects that are moving in it.
Now, how long have these initial devices been out there?
10 to 15 years for these devices, something like that. And progress has been slow. The devices don't change rapidly. The regulatory environment is complex, so it's not like there's all these big upgrades being performed. But one of the systems has had a couple of different versions.
That would be the Argus 2, is my understanding, is the most widely spread one.
Right.
And it's a few hundred people who have had one of these implants. Not thousands, but also not dozens.
Correct.
Now, at this point, there are many, many other devices that are in various stages of development throughout the world, maybe a dozen plus or so. Is that approximately correct?
Maybe we could say research projects trying to develop devices just like mine, as opposed to devices that actually exist and can be implanted in a patient. Yeah, there are something like a couple dozen projects around the world, people with different ideas about how to do things better.
Now, in order to understand how your approach is different from everything else that's out there, and it is the uniqueness of your approach that drew me to interview you, I think it'd be useful for many listeners if we could briefly review the basics of how light is captured and pre-processed in the retina before it's sent to the brain. That's really essential to understanding your unique approach. And the good news is, for non-scientists like me, it breaks down into 3 fairly discrete steps, each of which has its own distinct anatomical layer, only one of which I had personally ever heard of before I started researching all of this under your generous guidance, which is the rods and the cones, the photoreceptors, the input layer, in a sense. Could we start a little anatomy tour by briefly reviewing what photoreceptors are and what happens at that initial input layer of vision processing?
Sure. I think you said it exactly right. So the photoreceptors, the rods and the cones, are neurons. We can think of them a little bit like pixel detectors. They capture light over a very small area, and they transform that incident light energy into electrical signals, which is the currency of the nervous system. and then they transmit an electrical signal to a downstream neuron known as a bipolar cell and another one known as a horizontal cell. This is the beginning of vision because you have to translate light into electrical signals. That's what the photoreceptors do.
And so what's happening with all of these sensors, just to play back what you'd said a moment ago, they are picking up on photons, they're picking up on the quantity, the brightness, the color of light, the quality of light, and they're turning that into electrical pulses that they then hand into the second layer of our retina, which is, I'm going to just call the processing layer, which is bipolar cells and other circuitry.
Yes, I would only be careful about the word pulses. They don't send nerve impulses or spikes the way other neurons in the nervous system do. They signal information in an analog way.
Really?
Yeah. Many of the neurons in the retina primarily signal information in an analog way, and that makes the retina different from many other neural circuits in the brain. In many neural circuits in the brain, neurons communicate with one another with these nerve impulses or spikes.
Like it's binary, it's on or off.
Yeah, it's a momentary pulse, about a 1 millisecond pulse that happens at particular points in time, a little bit like a Morse code. Tick,
Yeah.
tick, 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 like that. The vast majority of neurons in the brain use spikes to communicate with one another. The photoreceptors communicate with their downstream partners using graded electrical signals, so gradual changes in the voltage of those cells and gradual changes in transmitter release from the photoreceptors to their downstream targets.
And how granular is that?
The granularity can be brought down to, well, what's the degree of noise? In other words, how big a distinction between a particular voltage level and another higher voltage level can you distinguish? So the answer is somewhere in the range of, I'm going to give you ballpark, 100 levels are effectively distinct in the photoreceptor signals, something like that.
Wow. So there is a very subtle signal that goes on to the processing layer. So let's talk about the processing layer.
The processing layers in the retina are complex and quite incompletely understood, even though there's been decades of research on the retina by very high-quality investigators. They fall into three major known categories, the so-called bipolar cells, horizontal cells, and amacrine cells. The photoreceptors communicate with the bipolar cells, and the bipolar neurons also connect up to the retinal ganglion cells. The retinal ganglion cells are the output neurons of the retina that transmit visual information to the brain.
And I've been studying this area with your help over the past several weeks. And during that time, I've personally come to view ganglion cells as being one of the most fascinating aspects of human anatomy. They're intriguing critters. Just before we get into them.
You and me both.
Yeah, oh, good. I'm glad they don't get boring upon familiarity. But just before we get into ganglion cells, I'd like to play back my layman's understanding of the first two layers. And please chime in if any of this sounds dead wrong. First, I view the photoreceptors as being kind of like pixels in that they're capturing tiny fields of raw light, and collectively, all those tiny fields stitched together to form the full field of view. Then the photoreceptors hand that basic visual information off to the second layer, which you just described, which does a bunch of complex processing, and that processing translates the raw data about the field of view into the languages of the ganglion cells. So that's the processing layer. Then these fascinating ganglion cells comprise the third layer, the output layer. So let's start off by talking about the number and the core function of these cells.
So there are about 1 million ganglion cells in each of your eyes, and those ganglion cells are the cells responsible for taking the output of the eye and transmitting it to the brain. They're very important in the sense that if you get rid of the ganglion cells, no signal goes from your eye to the brain, and so you will never see anything. So all of your visual experience, all of it, is ultimately encoded in the patterns of electrical activity of these 1 million or so retinal ganglion cells. These cells come in about 20 distinct types. The exact number of types isn't necessarily fully pinned down, but it's close to 20. Each one of these cell types is very distinct from the other cell types. They look different from one another. They connect up to different neurons in the retinal circuitry. They respond to light in quite distinctive ways. And they project their information. They send the output information to distinct targets in the brain.
And one of the things that's arresting about these cells, at least to a neural novice, is they're long. I mean, they're inches long. And the cell that has its input layer at the retina physically extends deep into the brain, correct?
That's correct. You have a neuron which is about 10 microns in diameter with an axon, the output cable, if you will, of the neuron that transmits its signal, which is less than a micron in diameter. And that tiny, thin cable extends inches into the brain to reach into these various distinct targets. They reach deep into the brain, as you just said, and they transmit these electrical signals into the brain where they are used by a variety of different visual areas for a variety of different visual functions.
Now, given that these cells are physically present deep inside the brain and also in the eye, is it fair to say that retinal ganglion cells are as much a part of the brain as they are a part of the eye?
I think of it that way. Some people say, well, wait a second, it's not the brain because it's physically a little bit distinct from the brain. But in fact, they behave just like other neurons. They send spikes. The spikes give rise to transmitter release at their axon terminals, and those terminals release neurotransmitter, and that affects the firing of the next cell along the chain, and so on. They're neurons like any other neuron.
They're inside the brain, speaking the language of the brain. And I guess an inverted way of putting it might be that, in a sense, the eyes are part of the brain.
I do think of it that way. Developmentally, the eyes develop from the same tissue that forms the brain. I like to think of it that the eye is a little outpost of the brain. It's the same kind of stuff. It's composed of the same types of cells with its own distinctive properties, but the same thing, and it connects deeply into the brain.
So let's talk about the fascinating diversity of the ganglion cells and the functional purpose of that diversity. You mentioned that there's 20-ish types of ganglion cells. My layman's understanding is that each one of those cell types is kind of like a team. Like the smaller teams may just have a couple thousand cells. The biggest team has hundreds of thousands of cells. And the teams aren't physically segregated. So it's not like there's one cell type at the top of the retina and another cell type at the bottom, but they're rather integrated with each other across the full surface of the retina. But although they intermingle, these teams are kind of independent of each other. Like each one of them creates its own distinct version of the field of view by stitching together the contributions of all of its own members. And those 20-ish different versions that result are really different from one another. They encode different information. They're sent to different regions of the brain. Is that a fair simplification?
Yeah, that's a very nice simplification. So the circuitry of the retina converges, if you will, onto these 20 or so different ganglion cell types. Those are the output neurons of the retina. They send the information to the brain. And the 20 cell types are really different in terms of the way that they represent visual information to the brain. So some cell types, as you said, there are many cells of that type, and some of them there are few cells of that type. All of the different cell types, as far as we know, convey a complete visual image to their particular targets in the brain. So for example, the very high density cell types will have many representatives that cover the visual image with very fine granularity and encode fine spatial detail in the image.
It's like they have more pixels. If your cell type has 100,000 cells as opposed to 1,000, you're going to have more pixels. It's going to be more detailed.
That's right. I slightly resist that analogy because I don't want people to think that they are pixels because they're definitely not. But yes,
right.
In a general sense, it's absolutely true that if you have more of them, you can get finer granularity on your image. Now, the different cell types convey different kinds of visual information to their particular set of central targets. So, for example, one cell type that's very numerous, has many individual cells of that type covering the visual image. They convey the finest spatial detail available. Another cell type conveys information about blue-yellow color vision, and that's a somewhat less dense cell type. Another cell type conveys information about rapid changes or flicker or rapid changes of any kind in the visual scene, and so on. And we don't understand exactly what all the different cell types convey, and what they convey is not easily boiled down into words. We can think of the different cell types as representing different Photoshop filters, if you will. Each filter is different. So if you take an image and you put it through this Photoshop filter, you get a sort of blurry, bluish yellow one. If you take your image and you put it through a different cell type, you get a different visual image. And each of these images is sent to its particular set of central targets. Now, what the brain does with all that is a quite open question. We don't really understand how the neural circuitry of the brain takes all of the outputs of these different Photoshop filters and pieces them together into our visual experience. If we understood that, we would be much farther ahead. Fortunately, for the creation of artificial retinas, we don't need to know what the brain does with it. Instead, what we need to do is make sure that what we deliver to the brain with our artificial retina basically resembles what the retina is normally doing. The correct Photoshop filter is being sent to the correct central location.
And then the brain can do all that incredibly complex and mysterious stuff that it does backstage, in a sense, so long as we're getting precisely the right signals that the brain should be getting in response to what is being seen at that moment.
That's right. That's the idea, that if we give the brain the correct raw material to work with in the format that it's expecting, that you'll get a visual experience that more closely resembles your natural visual experience.
Now, to give people a flavor for what the different cell types are like, without going through all 20 of them, could you give us a brief description of Parasol cells and what they seem to do versus Midget cells and what they seem to do? And I picked those two because they have the biggest teams. They're the most numerous cell types.
Actually, within those two, you can divide them up into two as well, so maybe we can talk about the four.
Sure.
There are so-called on and off Parasol cells and so-called on and off Midget cells. On cells of each category are primarily responsive to when something gets brighter, and the off cells are primarily responsive to decrements of light, something becoming dimmer. The Parasol cells of these two types have a slightly coarser representation of the world than the Midget cells.
So there are fewer of them.
There are fewer of them, and they're about a third as many Parasol cells as there are Midget cells. And they are exquisitely sensitive to change, to fluctuations in the light. So they sense that very rapidly and communicate it very rapidly to central targets. The Midget cells have a more fine resolution visual representation in them, and they are a little more capable of sustaining information about a static visual image so that you can keep seeing it. You're not just responding to transients. Those are some of the big distinctions between Midget cells and Parasol cells.
And to get to the heart of what makes your work unique amongst all the known teams that are working on devices to bring sight to the blind, it's that every ganglion cell type takes in a very distinctive code, or we could say speaks its own language. And you're deep in the process of decoding those languages. And uniquely, when your device talks to the ganglion layer, it's going to speak Parasol to the Parasol cells and Midget to the Midget cells. And in doing this, it'll replicate the job of that processing layer, which is where the distinct cell type languages originate in healthy retinas, correct?
So really all of this distinction in the different Photoshop filters, these different images that are sent out of the retina, are created within that complex intermediate layer in the retina that we spoke about before.
And what's fascinating is you do have this thicket of complexity, starting with the photoreceptors, which are relatively simple, and then into this processing material that basically ends up communicating a certain signal, let's say, to a given Parasol cell in response to the fact that I'm looking at a red wall. And if all of that complicated machinery were to die off, the photoreceptors and the circuitry in between, if somehow we could tell the ganglion cell what it would be hearing if only that stuff still existed, the ganglion cell, in theory, should be able to communicate back to the brain exactly what it would have communicated to the brain if the photoreceptors and the other stuff hadn't died off.
That's correct. That's how we think of the problem. Basically, the only signal that ever gets to the brain is the signal from the ganglion cells. The brain doesn't know anything about what these earlier layers in the retina are doing. All it knows is when the impulses arrive from the ganglion cells. So if we can reproduce in the ganglion cells the correct timing of the impulses they send to the brain won't know the difference. It receives impulses from ganglion cells. If you give it the right impulses from the right ganglion cells, it should do the same thing. That's the concept.
And this is critical because some very, very major forms of blindness, and specifically the ones that you're targeting, are essentially caused by the death of the photoreceptors, which means all of a sudden our pixels are gone, we're not taking in light information. And after the photoreceptors die off, there's sort of a slow, steady derangement that happens with that intermediate circuitry. It almost gets dementia. And that's what causes blindness in some cases. And that's what you could conceivably reverse by speaking directly to the ganglion cells in their own language.
That's correct. The two diseases that are targeted in these kind of devices these days are retinitis pigmentosa and age-related macular degeneration. The second disease being much higher prevalence in the population. In both of these diseases, photoreceptors die, and that's why you are blind, as you just said. However, the circuitry in the retina gets deranged. That was a good way to describe it. It's really very distorted and very unlike its normal, healthy state. But the retinal ganglion cells are still there. Their axons are still there. Their axons still terminate in the brain. And this is really what led people to say, wait a second, if we stimulate those ganglion cells directly, maybe those cells will send information to the right targets in the brain and people will see stuff.
So the ideal approach then would be to accept the fact that the input layer and the processing layer have died off or gotten deranged, and now start speaking directly to the ganglion cells, as we said, in their particular language. So what do you do to replace all that outside circuitry? So when you start talking directly to a midget cell or a parasol cell, how are you speaking to it? And in what language are you speaking to it? And how did you learn its language?
Okay, let's take one piece of that at a time, because that's a large one.
Big question, I know. Sorry about that.
These are great questions.
One question in 37 parts.
So the first thing is, how do we activate a ganglion cell? How do we cause it to send signals? And that is completely different from the way it normally gets activated, but it turns out to be okay. So what we do is we put an electrode close to the retinal ganglion cell, pass current through the electrode. That creates a little electric field that surrounds and encompasses the ganglion cell, and that electric field causes the biophysical mechanisms in the ganglion cell to produce a spike. Turns out you can basically force it to make one of these nerve impulses by creating an electric field outside the cell. Then the internal processes in the cell just march ahead and create an impulse because you've made it do that. That's totally unlike how it normally happens. Normally, the production of a spike in the ganglion cell is a consequence of receiving chemical input from the other neurons in the brain. So we're really hijacking it. And we're saying, forget about all that chemical input that you're getting from the bipolars and the amacrine cells. We're going to tell you when to fire a spike. We're going to put an electric field here right at this moment. It's going to be on there for 1/10 of a millisecond, and it's going to activate your internal mechanisms, and you're going to make that spike. So once you force a ganglion cell to produce a spike, the spike it makes is just like any other spike that it would have made. And it transmits to the brain just like any other spike that it would have made. So you're really fooling the system by jumping in and saying, I'm going to control when this cell produces impulses, and we're going to just ignore the fact that it's not receiving chemical input from the other neurons in the retina.
For all of the careful research I did in advance of this interview, I'm just becoming aware of what I believe is a very important and also fortunate nuance, which is it sounds like the ganglion cell takes analog-like input. It takes in very subtle, nuanced input, but its output is essentially binary. It either spikes or doesn't spike. And so you don't have to actually convey all that complex analog-like information inside the brain. You just need to know when to fire. And firing is either happening or it's not happening.
Yes, I want to be a little cautious. So conveying that slow fluctuating complex signal is what we don't have to do, as you said.
Yeah.
But we do have to make sure it fires at the correct times.
Exactly.
It's like a little Morse code. Click, 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 like that. You have to get the timing of that stuff right. We don't have to carefully control the voltage in the cell, which is hard. We just have to recruit the mechanisms in the cell that produce these spikes normally enough that they will just make one for us. So we just give it a pulse, even though that pulse is totally unnatural, it's nothing that it's ever seen before, that's okay. The cell responds to that by producing one of those impulses. So we just say, boom, we just made that cell fire a spike. We can control that extremely precisely. We can really nail when that cell fires a spike, and we can make it fire whenever we want to. So we can reproduce these little binary pulses with exquisitely precise timing. Actually, the timing precision that we can achieve with electrical stimulation is higher than the timing precision of the normal visual signal.
Really?
Yep.
Interesting. But of course, in order for the spikes that get sent into the brain to have any correlation at all with the seen world that you're trying to convey, you need to have figured out exactly when that particular type of ganglion cell would have fired in response to the scene that lies out in front of it. So I assume that you have done an enormous amount of very nuanced work figuring out when that complex chemistry that sits between the ganglion cell and the outside world would cause it to spike. You understand when a midget cell would spike as opposed to a parasol cell in this particular instance, given this particular visual layout in front of the eye.
Exactly. That's the basic research that everything that we're doing now is founded upon. You have to understand how the retina is conveying any given image into temporal patterns of spiking in a bunch of different ganglion cells of different types. You have to say, okay, normally it would have done the following. It would take that image and it would make these pulses at these times and send them down these ganglion cells to the brain. That's the basic science, figuring out how it normally works. Then we say, fine, got it. We have a model for how that works based on all the basic research that we have done and that many other laboratories have performed. We call it the neural code of the retina. How is it that the retina takes an image in and transmits it into a set of impulses that are sent to the brain? Once you know the neural code of the retina, now you're in a position to say, okay, good. It's my job now to reproduce that neural code by stimulating the cells and telling them when to fire.
If you want to be utterly comprehensive, you'd have to crack 20-ish neural codes because every ganglion cell has its own code.
That's correct. We have a rather good description of what the 5 major ganglion cell types in the retina do. These are the densest 5 cell types, so they have the highest visual resolution. And they form about 75% of all of the optic nerve fibers that carry the information to the brain. So it's a good majority of what's present in normal activity.
Classic 80-20 approach. You have found the relatively small minority that accounts for the majority of the activity.
That's exactly right.
Yeah.
So that's the position that we are in. Again, we are standing on the shoulders of research over several decades, but we've assembled that information using unique techniques that I think give us a much more comprehensive view of what that neural code is. So then our next step is to reproduce that code.
And is there a one-to-one correlation? Do you literally have an electrode going precisely to each of the ganglion cells that you're stimulating?
No, absolutely not. And that's where this all gets very interesting on the bioengineering side. You can't create a collection of electrodes that are custom tailored to every single cell like that. The biology is complex and idiosyncratic, as biology is, because we use devices that we can fabricate in nanofabrication facilities. We create a grid of electrodes, a fairly fine-grained grid that we place against the retina. When we put that grid against the retina, this electrode activates this particular cell over here. That electrode activates these 2 cells. That other electrode activates one cell, but a little bit on the other, and so on. So it's all complex, and each electrode activates cells of different types with different efficacy. So one part of the game to start with is, okay, given that our grid doesn't correspond to every cell, how do we activate cells in the way that we want to? If we want to make cell 1 fire and not cell 2 and 3 and then cell 4, how do we do that? How do we pass current through our electrodes in the way that causes those cells to fire and not the others?
And one of the secret sauces will be with all those electrodes, figuring out exactly what cells is each touching and which ones are we going to try to speak to, et cetera. And that is really the heart of what makes your approach different, not only from the couple of devices that are already out there, but as far as we know, the other devices that are under development is in conveying information to each ganglion cell in its own native code. Is that correct?
That's correct. And that is the defining central key aspect of our approach. So the existing devices that are out there, they don't work very well. And again, this is not to be critical of those really awesome efforts, but they don't work so well that you would give up your guide dog or your cane. So what's wrong? Well, we don't know for sure, but the hypothesis is going and stimulating with a grid of pixels is just not good enough. Why? Because there's all these different cells sending different signals to different targets in the brain at any given location. You're just activating pixels in a grid and hoping that the wash of activity you create with those pixels is going to be somehow interpretable to the brain. We don't think so. We think the brain is really precisely wired up to receive input from different cell types and interpret them in different ways. So if you activate the wrong cells to fire spikes at the wrong times, the brain gets really confused and you don't see very well. The analogy I like to use, that I think I've used in our discussions before, is that the retina, with its many different cell types, you can think of it as an orchestra trying to play a symphony. The different instruments have different scores. They should be playing at very particular times. The scores are different from one another. And if you want to create that symphony and you really want to make music, you need to get the right cells to play their notes at the right times. And you don't just want to stimulate all the cells and make them play at the same time. You can raise your wand and conduct in a way that pays no attention to the different instruments. You can give them all the wrong scores and the wrong sheet music. What are you going to make? Probably not music. You probably make a cacophony. It's audible. You might even be able to recognize the tune in the background there if you listen carefully, but it's not going to create the experience that we want to create. We want to conduct that orchestra to play a piece of music. And in order to do that, we got to pay attention to what the different instruments are and when they should be playing their particular notes. We've done a lot of research in the lab that shows that we can stimulate individual neurons, individual instruments, if you will, to play their particular notes. We can actually do that very accurately. But you can't do that if you don't even know where the cells are and which electrode is close to which cell of which type. If you just have no idea which ones you're stimulating, you have to pay attention to that.
It's like giving the clarinets and the oboes and the violins all the same sheet of music in a symphony as opposed to their own unique scores.
Right. And you can do something with that. I don't want to make it sound like that's useless. You can play chopsticks with an orchestra, right?
Yeah, you could.
It's fine. And
Yeah.
that's a terrific start. That shows that there's something to be done here. But that's kind of where we're at right now because we don't pay any attention to the neural code of the retina. So our fundamental hypothesis that guides everything we're doing in the lab right now is that you have to actually speak the language of the brain. You have to send information that the brain can actually unpack in a reasonable way. And in order to do that, you gotta tell the different cell types to send their impulses at a particular set of times that reasonably closely matches the way it is in the healthy retina.
And again, what I love about this approach is it allows the brain to do its own thing. It would be wonderful to understand all the wildly complex things that are going on in the visual cortex once this information comes in from the ganglion cells. It may take us centuries to figure that out, though. For now, you simply got to get those ganglion cells to fire when they should fire, and the brain will do all the rest. And this is just so much more of a tractable problem than figuring out everything that goes on inside the brain when it's creating a visual world.
Yeah, that's absolutely right. And that's what we're leaning on. In the interest of being very careful about what we know and what we're still figuring out, raise a little warning flag to watch out for. In the process of losing sight, when the photoreceptors stop working and the human patient is unable to see, who knows how things get rewired in the brain?
Right. On the inside, we don't know, do we?
We don't know. And could it be that during that process of losing sight, some of the mechanisms in the brain also degenerate in a certain way? Yeah, that could be.
Right, because that's going on in the bipolar cells. It's going on in the outer circuitry, so it may be going on in the inner circuitry as well. Very interesting.
That's right. So we don't really know, and that's probably the case that different degrees of central degeneration occur in different patients, and that may explain why some clinical outcomes are better than others in the existing devices. There's actually a huge variation in the quality of the clinical outcomes. So we don't know for sure. What we think is true is if you did this in a healthy retina, you would never do that. But if you did that in a human healthy retina and you plug in your device and you reproduce neural code, we think that you would see basically exactly what you would see in healthy vision.
Because there would have been no degradation on the inner circuitry, yeah.
That's right. That's what we think. And so what happens after many years of degeneration? We're going to have to find out.
I interviewed Mark Humayun for the same New York Times article that I interviewed you for several weeks ago. And for listeners, Mark Humayun is the co-inventor of the Argus 2. Is that roughly correct?
That's roughly correct, yeah.
And he had told me that they don't even put the Argus 2 retinal implant into patients unless they are people who used to see and lost their sight. They don't even put the Argus 2 into somebody who has never had sight because they're concerned that that underlying neural wiring simply won't be there. I don't know if they experimented with that and just had poor results with people who had been blind for their entire lives. But my understanding is that the protocol now is that it only goes into people who lost their sight.
Yeah, very unlikely that they even bothered to experiment. If you've never had sight, there's no way that your visual brain is going to be in a position to make sense out of what you introduce with one of these artificial devices.
So let's talk about your anticipated initial device. I think you told me 4 to 5 years out?
That's the goal.
And I think you said it'll probably be 5 of the 20-ish ganglion cell types that you'll be speaking to.
That would be our first target. That's correct.
And characterize the type of vision a patient would hopefully obtain in terms of field of view, resolution, color, etc. What do you expect the experience to be like if all goes as hoped?
Yeah, great question. What we expect to be able to do that's very different from existing devices is to create a rather high-quality visual image. It won't be your entire field of view by any stretch of the imagination. It will be limited by the size of the device, and we're still discussing exactly how big that would be. But you might imagine an image that's about the size of your cell phone, held at arm's distance out in front of you, the way you might hold your cell phone. Some image like that, but it's a pretty good quality image. You can actually recognize features on there on a device that big. Something like that is what we're targeting.
The field that's essentially illuminated by sight would be roughly the size of a cell phone at arm's length. So it'd be kind of like shining a very tight spotlight around a dark room, maybe?
Yeah, that's a nice way to view it. Mm-hmm. Existing devices actually cover a larger field of view, but with very irregular, and as we discussed before, poorly controlled blobs of light that are huge and don't make a whole lot of sense in terms of what's being activated. We think that by properly activating the different cell types and replicating the neural code of the retina, we can create that reasonably high-quality visual image. We may be able to create color sensations. we will see what we can create. We know what we're going to do. We're going to sit there and grab onto those 5 major cell types as our starting point. We're going to reproduce the signals as accurately as we can in those cell types using the architecture I described. And then we're going to see, what do patients observe?
And it will be reasonably high fidelity. Do you think people will be able to read within that field of view?
Quite possibly. And experiments have been done showing that people can do some reading. Reading is a task that involves not just seeing an image momentarily, but of course tracking your eyes across the page.
Right.
And so learning how to track that spotlight, if you will, across the page is something that people will have to learn how to do. If the spotlight is a little bit off the center of gaze, that's a little bit harder, but it's still doable. So there's stuff to work out with that. But yeah, the hope would be that you have sufficient resolution. Each time you flash that spotlight, you can see a word.
And how much of the total system will be implanted? Obviously, the electrodes will be implanted. Will there be an external element as well? Would somebody wearing this device look like they're wearing glasses?
Exactly the right question. Our calculations and thinking about how to design the correct interface tell us that what you want to do is to have a device that's implanted, surgically implanted, that is lean and mean and minimalist. It dissipates as little heat as possible so you don't overheat the retina. It's not complex, it's not bulky, and perhaps most important, since it doesn't have the complexiness and the bulkiness inside the eye, and some of the stuff is therefore on the outside of the eye, things can be upgraded and improved over time.
The exterior hardware very easily.
Right.
Yeah,
So,
non-invasively. Let's say you launch with 5 of the ganglion cells understood, and as time goes by, you crack number 6, you crack number 7, 8, 9. You might be able to actually add those new cell types to a given user's device by upgrading the software?
absolutely, because the software will be contained in your glasses. So we give you a new pair of glasses to get cell type number 6. That's exactly the idea.
It'll be fascinating as you do this work to see what those 5 major ganglion cell types enable. I'm sure you will have intuitions and instincts about what cell type number 7, 8, 9, 10 are going to add. But then you're going to find out factually, like, who knew? Cell type 13 is where flicker lives,
Right.
all flickering things. Now they can enjoy strobe lights, and we never knew it was going to be cell type 13.
Yeah.
But that'll give you incredible insights to what these different cells do.
That is exactly right. And so let me say 2 things in response to that, because you raise all the right questions. When you stimulate all the cell types indiscriminately, when you try to conduct the orchestra without regard to which instrument is which, you create conflicting signals. You create information coming from different cell types that is mutually incompatible. It's a short circuit for the brain. It's like, this doesn't make any sense. This cell is telling me that it's getting bright. This cell is telling me that it's dark at the same location at the same time. What's going on here? Right? So you create a conflict with these different instruments doing the same thing.
Oh, that's interesting. So if one electrode is, in effect, speaking within the hearing range of 2 ganglion cells, which each have their own languages, one of those cells could get very confused, especially if a certain word means different things in different languages. Like, I happen to know that the English word gift, those phonemes, that raw sound, gift, happens to mean poison in German. So if you went up to a German and an American and held out a little package and said gift, the American would think you're being generous, and the German would think you're trying to kill him. Is that what it's like when an electrode speaks one language to 2 different ganglion cells?
Yeah, and it's more extreme than that. It's more direct than that, and because there's so-called on cells and off cells that signal increments and decrements of light.
Right.
If you stimulate them at the same time, what you're telling the brain is this: at this location, it's bright. Oh, and by the way, at this location, it's dark. It's bright and it's dark. Our visual systems don't have any way to interpret that. It doesn't make any sense. It can't be both bright and dark, right? So coming back to this issue about all the different cell types and whatnot, is I think a precise, fine-grained stimulation of just one cell type would probably be a lot better than an indiscriminate activation of many.
Ah, right.
This is a hypothesis. So in fact, the first thing we'll try is one cell type. That will be the very first thing we try. Can we make a decent image in one cell type? How about 2, 3, 4, 5? We will add to them and see what we can do, and then we will work on the others in the future. So that's point number 1. Point number 2, I think, is this question about what do the different cell types contribute to our visual experience is something that we have not really been able to study in all the basic science research. Why? Because we don't have control over the different cell types. We can deliver a visual image to an experimental animal or a human and interrogate their nervous system, but we can't put our hands on the handlebars and take control over the signals. With this device, we will take control over the signals. We will be able to dial up what this cell type is doing, what that cell type is doing, what that cell type is doing. We'll be able to try just cell type 1, cell type 2, both cell type 1 and cell type 2. What if we mess around with the code in cell type 2? What I'm trying to say is that on the way to creating this clinical device, we will create what I think will be a spectacular research instrument.
And you'll also have subjective reports coming back from people who are mentally experiencing exactly what is happening. And that's something you could never get from an electrode or any kind of animal, et cetera.
That's right.
You'll have somebody being able to communicate in highly, highly nuanced language exactly what's going on. Now, one of the most fascinating aspects about this work that I became aware of when I started digging into it, again, for that New York Times piece that I first interviewed you for, is that there's a very fine line, and indeed a fascinating line, between therapy and augmentation. And the Argus 2, the certainly breakthrough, but nonetheless relatively simple device that's already on the market, already enables its users to see infrared light, which is something that no other human can do without the aid of night vision glasses. And the reason to enable that, Dr. Humayun told me, was to prevent thermal injuries. If somebody's going to be able to use their Argus 2 to navigate their home in a more intuitive way than they could before, if they've got a burner top on top of their stove, they may as well know that that thing is 400 degrees, and infrared data will show you that, whereas our simple eyes will not. Would you care to talk a little bit about the potential for augmentation even when you're in the early therapeutic steps? What are some of the visual, perhaps superpowers that you could bring to somebody who was until very recently blind?
Yeah, that's a fantastic question. You actually took my example, so I can't use that example.
Oh no, sorry about that.
No, not at all. But let me just go a little further with that and then comment on other possibilities. We often think about a distinction between restoring normal vision and augmenting vision. In fact, the funding agencies think about that. The National Institutes of Health will not fund research on augmentation. They'll only fund research on restoring vision. So people try to draw that bright line. Once you've developed an implant of this kind, to restore infrared vision is a matter of literally removing the infrared filter from in front of your camera. And then all of a sudden you got infrared vision, just as Humayun said to you. So in my view, there is no bright line between augmentation and restoring function. It's an incredibly blurry thing. You're there before you know it. Now, what will we do beyond that? Okay, so now we get into a very speculative and interesting domain. Let me give you one simple example, which is fun to think about at least, even if it's not necessarily the most realistic. You know that we can drive a car and have a phone conversation with earbuds or hands-free phone conversation with reasonable safety. Let's say it's not quite as safe as not having the conversation, but it's not too terrible to the degree that our laws permit that. On the other hand, trying to drive a car while reading text is a disaster and extremely dangerous, right? So our laws don't permit that. Okay, so why is that? Because our auditory language system, which is what you're using when you're having your conversation, is very different and sort of parallel to our visual system, which is what we're using to navigate and make sure we don't crash into another car. So you have two systems that can work very well in parallel and not really interfere with one another very much. And that's why we're able to be reasonably safe having those conversations. Okay, now could we do that in the visual system? Could it be that if we were able to harness the different cell types in the way I just told you about, and control this cell type just like we want, and that cell type just like we want, and so on. This is a bit tongue-in-cheek because I'm not planning to do this, but could we, for example, introduce an image of the oncoming road and visual environment and the cars on the freeway into your Parasol cells, and simultaneously introduce a totally different image of some words into your Midget cells, different cells, project to different areas in the brain, and be able to safely read while driving? That's possible.
Because you'd have totally different regions of the brain doing the two things and not interfering with each other. You wouldn't be getting words of the car and you wouldn't be getting wheels and highway in the words.
Potentially. And again, this is more or less a tongue-in-cheek example. It's not like this is our purpose to do this, but just as a way to think about the problem. But what we will try to do is introduce conflicting signals in different cell types that could interfere with one another unless they're separate. And if they're separate, that means you now have an ability to do two different visual things at the same time.
The first thing that somebody might think of would be a heads-up display. Okay, I'm looking at my windshield and I'm seeing the oncoming traffic, and I'm also seeing today's news. But this would be a complete separation, not a mere heads-up display type of experience.
That's right. We would be creating a visual image that you have never seen before. And it cannot be created by that. Why? Because that thing is literally superimposing characters of text on the image of the road.
There's one image that's got two things. Yeah.
You got it. And all the cell types are responding to that image the way that they always do. So the Parasol cells are seeing that image of cars plus text, and the Midget cells are seeing that image of cars plus text. We're saying, no. We're going to put the cars into the Parasol cells and the text into the Midget cells. That can't be done with a visual display.
No.
It's not possible. You can only do that if you take control over the cell types the way we're proposing to do. Now, again, that won't be our actual thing that we're going to try to do, but we will try to experiment with how independent are the different channels of visual information. Again, we'll be able to do that early on in experimental animals to see whether animals can track two different things at the same time. And then eventually in humans, where we ask people, are these particular sensations we introduced in some sense independent of one another? And could you therefore use them separately? Why would we even care about this? Well, in some larger sense, what we're doing with a device like this is tapping into an incredibly high bandwidth information corridor into the brain, or many corridors coming from the many cell types. We are harnessing that. We're able to plug into these many ganglion cells, sending many signals to many targets in the brain. We may be able to introduce visual images that are richer and more complex than any normal sighted persons can ever see, so that you can, for example, maybe walk and read at the same time, or have a conversation with somebody and recognize their facial expressions while navigating your environment safely. Give yourself not just one visual system, but 2 or 3 visual systems, let's say. Content, higher bandwidth delivery of visual information into the brain by harnessing these parallel channels.
So new types of multitasking, in a sense, could be possible. The reason that we can walk and carry on a conversation at the same time is that they're very, very isolated activities, and indeed geographically isolated within the brain. If we were able to start treating vision that way, it could enable, A, exotic forms of multitasking, but B, also, of course, you could introduce, by accessing it wirelessly, et cetera, outside information that you would have no access to with your eyes or with your brain or with your body.
That's right. That's exactly right.
Now, when I think about the near-term forms of augmentation, I imagine that just like Mark Humayun with the Argus 2, when you deliver your device, you will probably enable some kind of infrared because it is useful. It can avoid thermal injuries. And by the way, it can give somebody night vision, which is something that I've sometimes wished that I've had. Is that a likely augmentation that would be present in the initial device as it goes out?
Entirely possible. Once we provide the ability to control the different cell types, this question of which information do you provide to which cell types at which times is easily controlled by software. Anybody who knows how to write Python code or whatever could take different visual images and introduce them to different cell types in different ways. In other words, we're sort of creating a software developer toolkit. We're saying, here's the toolkit. Here's how you take control of those different cell types. Go to town. Try different things. In much the same way, you have app stores where people develop apps. Once you develop the enabling technology, then people will try this and they'll try that, and creative ideas will come up about how do you merge text and image of walking and beautiful pictures that you look at behind the scenes and facial expression of the loved one. How do you mix all those into your visual sensations? Hopefully people will have ideas about how to do that once we make it possible to take control of this visual information channel.
And so much as Apple, for all of the brilliance that was resonant within that corporation when they shipped the iPhone, they couldn't hope to think of even a tenth of all the powerful things an iPhone might ultimately do. It sounds like you would be open to taking a similar approach and literally making an SDK, a software development kit, available to people to figure things out on their own. I mean, there's a lot of brilliant postdocs out there and graduate students out there that could probably come up with amazing notions that wouldn't necessarily occur to the small handful of people in your lab.
Exactly. We need to build the instrument first and then unleash the creativity of many people to augment our capacities by tapping into our visual system. And there will be people who have better ideas than I do on that, for sure.
Now, when you think of the device that might be shipping, let's say 10 years from now. So let's say if everything goes well, you get approvals in 3 to 4 years. It might be out in the marketplace, 5-ish years from now, conceivably?
That would be nice if the regulatory approval goes quickly.
So if there's some good regulatory tailwinds, let's say 5 years it's out in the market, maybe 5 years beyond that. So you're at generation 2, 2.5, 3. Do you think at some point people might start doing this electively. People who have perfectly good vision might start saying, hey, you know what? I'd like to have this in my peripheral vision. I don't want it in the middle of my fovea and the dead center of my vision, but in my peripheral vision. Do you think people might start doing that?
It's a brave new world, isn't it?
Yeah.
First, why don't we ask, well, why not? Well, why not is because you don't want somebody cutting into your eye to implant something and all the consequent hazards and risks, right? So if the surgery is sweet and clean and goes well, and it's done many times over, and it's a really good, straightforward approach, people will be more likely to adopt it. If our surgery works well and our implant is robust and doesn't cause problems and gives you a capacity to see something that you never saw before, that you never could possibly see, will some people be interested in that? Yeah, I think so. Those are the early adopters, right? The more powerful we make it, if we show that there are some real breakthroughs in terms of encoding simultaneous information in different ways, or maybe it's as simple as something that is stunningly beautiful. Maybe we can create beauty that's just unimaginable from what we normally experience, that there's just extremely appealing images. I don't know. Again, this is where creativity will come in. Do I think augmentation will happen on a voluntary basis? Absolutely. Will it be from our device or from a future improvement? I don't know. We're going to make our device as good as we can.
Well, it's fascinating to think of what an artist might do with this. I mean, if you get the SDK, as you call it, down to a level of abstraction and non-technical accessibility that something like Lightroom is or Photoshop, those types of software, artists access that and do amazing things. Once the 20 cell types are very well understood and the software and the interfaces are well understood, it's very easy to imagine that artists could create spectacular visual experiences that are either filters on top of the existing world or perhaps entirely virtual experiences by twisting these dials in manners that mere podcasters and scientists may not think of.
Yes.
I hadn't thought about that, but yeah, the potential for an unleashing of an entirely new type of beauty and a new type of art is pretty intriguing. Now, another thing that's intriguing to me about this is it started sinking into me that ganglion cells are as much part of the brain as they are part of the eye. I started realizing that the work that you're doing is perhaps an alternative to neural implants. And we have people starting to talk about perhaps consumer-grade neural implants, companies like Neurolace, which Elon Musk started up, companies like Kernel. And whenever I hear about neural implants, I get worried because it sounds an awful lot like somebody's going to crack open my skull and insert some metal into my brain, which I don't know why, maybe I'm just a wimp. Sounds scary. Now, obviously, a retinal implant is no minor thing, and you're playing around with sight and so forth. But once you get that to the point that it is a common procedure, like a cochlear implant, that sounds a lot less frightening than inserting metal into my brain. And because you are so intimately communicating with all kinds of depths and regions of the brain with these ganglion cells, do you think that the approach you're developing might become an alternative to neural implants? I mean, obviously not for things like helping Parkinson's sufferers not have seizures. That's a very different thing. But in terms of sort of the life-augmenting aspects or even intelligence-augmenting aspects that people hope to get from neural implants, do you think that the eye may be a different way to achieve that?
I'm glad you raised that question because I think about that kind of stuff a lot. Yes, I think for the domain of visual experience and acquiring information about your world through sensory input, I think the eye is a way better way to go than the brain for a couple of reasons. One is that cracking into your skull, and it's something you want to avoid. Also, we actually understand pretty well how the retina works, whereas what happens in those brain areas is much less well understood. So we're way ahead of the game in terms of being able to interface effectively to the eye compared to sticking electrodes in the brain. As you said, there are companies that are working on electrodes in the brain, more power to them. That's also a good parallel approach. But in terms of visual information, you're way better off on the retina. It's not even close. So that's number one. But I also want to put on the other side, there are categories of neural implants that we're not going to touch. The eye is not used for memory storage. If you want to do memory augmentation, that's going to be an implant into the memory areas of the brain. If you want to do communication and language in a direct way, and you want to make it so that you provide new augmented capabilities for humans to communicate with one another in a very fast and transparent way.
Like telepathy type of things.
Essentially, yeah. So we're really now in science fiction mode, but
yeah,
it's not that far off.
Right.
where we could envision implanting devices that read from the language centers or write to the language centers and thereby allow people to communicate or allow people to just process information more rapidly. So there's a whole bunch of stuff that we won't tackle with the eye and that you need brain implants for. Leaving aside which particular thing we're tackling, there's a fundamental problem that's common to all of these neural interfaces, which is that what you're doing is creating a grid of electrodes that's manufactured in a foundry, sticking it in the brain, and then starting to communicate. And I think this problem that I emphasize in the retina about the many different cell types doing their own signaling, this is going to be common to other areas of the brain as well. As far as I know, it'll be the ubiquitous finding that you can't just stick a grid of electrodes in there and pass current and hope that something good is going to happen.
Yeah.
You actually have to fine-tune your device, calibrate it, have it adjust, and figure out what's going on around it to communicate with the local neural circuitry. We are figuring out those problems now in the retina. We're able to do it because the retina is more accessible. We have a lot more knowledge about the retina, and we've been doing these kinds of large-scale recordings and stimulation of the retina for years. And I think we will solve those problems in the retina well before we solve them in the brain. And the ideas, the approaches, the technical machinery for doing that, the intellectual property for doing that, we will develop in this process, and it'll be highly applicable to other brain interfaces of the future.
And again, I keep getting back to the relative tractability of the retina. I mean, you're wrestling with 20 neuron types. It's not easy. You've spent many years at it. You're getting there with 5. You haven't got all 20 conquered. You don't even have all 5 conquered. But it's only 20. How many neuron types are in the brain?
We don't know.
We don't even know.
That gives you a sense of the problem. We don't even know. People argue about it, whatever. I'm going to guess that in the primary visual cortex, which is the major recipient of visual information, the biggest one in the brain, I'm going to guess there are a couple hundred different cell types. I think it's a reasonable guess.
And presumably, if what happens in the retina is analogous to what happens in the brain, we might safely assume that each one of those neuron types is going to have its own distinctive code that needs to be cracked independently. And they're going to work together in a synergistic manner that is, in many cases, I imagine if you get into the realms of cognition or consciousness or memory, they're going to interact with each other in ways that don't have these relatively accessible metaphors, like think of it as 20 different Photoshop filters. I think metaphor is actually very, very important. We can wrap our heads around that and start having an intuitive sense for what these 20 different neuron types do. But if we get into the hippocampus and discover that there's 650 different types of distinctive neuron that are involved in memory, we don't have any kind of simple way of just not navigating that conceptually. That's a hairy problem.
It's a super hairy problem. You put your finger on it beautifully, and you said it exactly right. We don't even have the right metaphors. We don't even have the right ways to think about what the different cell types are doing. People are working on that, smart people, and so they're going to make progress. But it's going to take time to figure out what all those different cell types are doing in the brain. And I don't want to make it sound like, oh, we retina people are so smart. It's not that we're smarter. It's that it's easier. We figured out in the retina because it's relatively simple. It's a Photoshop filter, something we can wrap our heads around. And it's a one-dimensional sheet, so we can study it. And it's easily accessible. That's why this field, in terms of knowing how the retina works, is decades ahead of knowing how the brain works, not because we're smarter, but because it's easier.
And that accessibility is no small thing. I mean, if we do accept that the eye is part of the brain, it's the part of the brain we can access. It's the part of the brain we can actually look at. And it's also been studied much more deeply over more decades, right?
Yeah, exactly. And more progress has been made because of that. In fact, there's a textbook that I believe that the textbook's second line of the title is an accessible part of the brain.
Oh, interesting. That's great. To close on the super speculative stuff, you have mentioned in passing enhancements to memory, enhancements to other aspects of cognition, other aspects of what the brain does. Where do you see neural augmentation going, let's say, in the next 25 years? Do you think we're going to see really awesome breakthroughs in areas like memory or other areas that you might have deeper insights into?
It's pretty hard to say. What's safe to say is that the areas which are best understood on the basic science front will be the areas that we augment first. What are those? Well, vision, audition, as we already talked about, motor control, and people are working on devices that allow you to control cursors, and robot arms are already being controlled, artificial limbs.
Merely by thinking about it, quote unquote.
Merely by thinking about it, that's right.
Which is incredibly life-enhancing for people who have lost some motor control or even worse, are paraplegic or quadriplegic.
Exactly. And then the same issues come up in terms of the relationship between therapy and augmentation. So right now we see the spectacular capability of helping people who are profoundly disabled. That's great, and that's what's happening. So in the future, once those implants become safe and routine and they're well tolerated and everything, if you have a high bandwidth output from the brain, let's suppose you want to control a very complex fighter jet of the future.
Right.
Rather than use your hands, your 10 digits that you move around as quickly as you can to affect different controls and whatnot, what if you just have a very high bandwidth readout that this is what you want to happen with all the different instruments? Can you do it way faster and to control a fighter jet that's way more complex than we can now? Or another piece of machinery, construction machinery, or your computer. You literally want to write code in a really fast way. Can you develop ways to read out signals through the motor system that you can imagine controlling your environment, whether it be keystrokes or instrument panels or whatnot, in a way that's much richer and much faster and gives us much more high bandwidth capabilities than now? I think that will be the other obvious frontier, motor control.
That motor control stuff is intriguing, and when I see videos of it online, it looks very much like magic when you see somebody controlling an external arm simply with their brain or even a cursor. And is the most sophisticated and nuanced stuff that's happening in motor control right now, is that through implants, or is it through reading electrical activity out through a headband or something?
Both, actually.
It is both.
Yeah,
Interesting.
both. I mean, non-invasive ways are pretty good. I would say that the motor implants at the moment are not a whole lot better than what you can read out with non-invasive techniques. There's not a dramatic difference, but the basic science is being worked out so that hopefully soon there would be a dramatic difference. But I would say that the day isn't here yet where that's the case. Both are really important, and non-invasive obviously plays a huge role. Back to your question, what other areas will there be? These are the more speculative areas. I think they're further off because we don't understand them as well. So like memory implants. There have been speculation about memory implants. I'm sure that will be eventually an awesome target. We don't really understand how the memory system works well enough to build a device to augment that system at the moment.
Yeah.
A lot more basic science is needed, so that will come later. Communications devices for interacting between people. Again, we don't understand it deeply enough that we know how to build something that's going to work better than what's already there. It's only in these relatively straightforward things, sensory systems and motor control, that we can imagine augmentation.
Well, it's an amazing realm of possibility. How do you think people are going to react to this sort of thing once it becomes widely available?
This stuff is spooky, right? The idea of implanting something in your brain to control different devices and communicate with people, this stuff is spooky, it's weird, and many of us may not want it. How will that change in the future? Well, I think younger people will be more open to the technology, as they always are. They'll be more likely to adopt it, as they always are, particularly if it's well worked out surgically. And what does it mean for humanity? What I like to think is that it's an enabling technology, that we will be able to think better, sense better, remember better, control things better, communicate with one another better, and that will allow us to solve larger problems that humanity faces. Others will say, this is crazy. Why would you want to start introducing devices into your brain? It will be used for all sorts of terrible things. People will hack into your brain and control your mind. People will be concerned about that. Rightly so. Any technology that's really transformative brings up possibilities for use for good and use for evil. There's no question. Every single technology is like that. The upside, however, is spectacular. And if it allows us to think, communicate, sense, move better than we have before, maybe we can tackle some of the challenging problems that humanity faces better. Maybe rather than wait for tens of thousands of years for our genome to change and for us to acquire better capabilities, maybe we will evolve much more rapidly by merging our capacities that we dream up and build with our bodies. And if we can show this crucial idea, the central concept that we need to reproduce the neural code, if that makes a dramatic difference and all of a sudden we start being able to create good visual images, we will scale this thing up. That won't be super slow.
Access to talent and capital would not be a problem once you showed the initial proof of concept that this is really working.
Well said.
Yeah. EJ, thank you so very kindly for your time this afternoon. And it's brilliant stuff, and it's an amazing education as well. So thank you for all of that.
Rob, it's been a great pleasure, and the questions have been extremely interesting. I really enjoyed it.
Thank you kindly. I just love EJ's work. Among other things, you can't help but learn a ton of neuroscience by digging into it, both as a listener and definitely as his interviewer. Now, I have a lot more to say about my conversation with EJ in a moment, but first, I want to give you a somewhat playful preview of my next episode's subject later this month. What you're about to hear is a brief recording made this past Friday between air traffic controllers in Ireland and multiple commercial aircraft that were flying in the vicinity of Shannon on the western edge of the country. Here we go.
American 86, reflectable 370, and it is smooth again.
American 86, Shannon. Hello, radar contact, and it seems to be smooth ahead of you as well.
134.26 is Singapore 25 confirmed. Paper.
Meteor or another object making some kind of re-entry.
Appears to be multiple objects following the same sort of trajectory. Very bright from where we were.
Okay, that's copied. And is there a direction it was going in or anything?
The Virgin 76 also saw that in our 11 o'clock position, two bright lights.
Roger, that's copied. Thank you.
Glad it wasn't just me? No, yeah, very interesting, that one. Virgin 76, I saw two bright lights, 11 o'clock, seemed to back over to the right and then climb away at speed, at least from our perspective.
Okay, we're passing that on now. Thank you.
Speedbird 94, Shannon.
94, go ahead.
Okay, just so you know that other aircraft in the air have also reported the same thing, so we're going to have a look and see.
Now, I edited out some pauses and a couple of voices that were hard to understand, but that was the raw recording from the headsets of the pilots and the air traffic controllers above and around the west coast of Ireland on Friday. And I can tell you with absolute certainty, those pilots did see a UFO. Because, of course, the U stands for unidentified. And for now, and probably forever, nobody knows what those rapidly moving objects were. But I can also tell you, with almost as much certainty, that the pilots did not see an alien spacecraft. Because decades of careful and level-headed research indicate that they probably saw meteorites. And if not, it was almost definitely something way more boring than Little Green Man. So like I said, that was a bit of a playful setup for a preview of my next episode, which is on a somewhat similar mystery, only it's much deeper, it's far more controversial, it's unique in human history, it's unfolding right now, and on a much vaster scale, which is to say, our solar system, and indeed, far beyond it. The mystery surrounds an object called Oumuamua. It was first sighted retreating rapidly from the sun in October of last year, so about a year ago. And it's the first object ever sighted in our solar system that astronomers can say definitively came from outside our solar system. Now think about that. We've been doing astronomy for thousands of years, and we've been doing it with really sophisticated gear for almost a century. We've identified countless asteroids, meteorites, comets, and other things. And this is the very first interstellar visitor that we've known about. Oumuamua was tracked very closely by teams throughout the world, and also by the Hubble telescope. It had several strange attributes, which we'll discuss in our next episode. A biggie is that as it retreated from the sun, it started accelerating in a fairly strange pattern. And then in January, it faded from sight on its long journey out of the solar system. By this past summer, the consensus among astronomers was that Oumuamua was a comet of some sort, although a remarkably strange one on multiple levels. Then on Monday of this week, Astrophysical Journal, which is a very highly regarded peer-reviewed academic publication, published a competing theory. The new analysis, which is deeply controversial, suggests that Oumuamua was no comet. Rather, was a very small object, roughly 20 meters in radius and less than a millimeter thick. Now, no known natural phenomenon could produce a spaceborne object with anything approaching those dimensions. The authors of the paper compare this odd shape with a solar sail. Solar sails are a proposed method of interstellar spacecraft propulsion, which Japan's space agency has experimented with successfully, and which a well-funded private initiative to send spacecraft to a nearby star is now researching intensively. So yeah, an actual alien artifact. And the paper's lead author doesn't have the job of a crackpot. Specifically, he's the chairman of the astronomy department at Harvard University. His name is Avi Loeb, and he'll be my next guest. Avi first put his paper online at the same time he submitted it to Astrophysical Journal just a couple weeks ago. The journal accepted it in 3 days, which is extremely unusual, but it was still sitting out there online. A couple of smart blogs picked up on it over the next few days, and then about a week ago, it started breaking into the mainstream press in a really big way. And I'll repeat, Avi's ideas have been very controversial within the astronomical community. I mean, there's some major vitriol out there. Now, these sorts of debates happen all the time. Anomalous findings are discovered, radical explanations are proposed, brawls break out, sometimes they last decades. This is actually one of the key dynamics that drives science forward. And the debates often get very heated. And I think this adversarial process of analysis and discovery is worth exploring on this show, along with the less controversial cutting-edge developments I usually bring to you. And I thought this debate would be an especially interesting one for all of us to explore because it's happening right now. The credentials of the heretic in the conversation are really impressive. And let's face it, it's about aliens. And I'm a science fiction writer. Now, I will in no way try to persuade you that a Oumuamua is an alien craft. And I actually don't think Avi will try to persuade you of that either. I think his goal is rather to make us all much better informed and open-minded as this conversation unfolds, as it probably will for years. I'll add that alien technology has been considered as a potential explanation for multiple strange phenomena in astronomy over the past decades. In almost all cases, it's ultimately been ruled out. But that's always on a path to interesting and fundamental discoveries about how our universe works. And I believe those tantalizing moments or years when the A word, that's alien, is uttered by serious thinkers who are considering a mystery can excite a lot of young people to tune into science and astronomy and sometimes to fall in love with it. That actually happened to me when I was a young kid. I'll probably tell the full story in the next episode, but the cliff notes are that something which turned out to be farcically non-alien briefly seemed really spooky to some astronomers. I got very excited about that story, and though it turned out to be nothing, my fascination with astrophysics has never diminished. Avi Loeb has been overwhelmed with press attention in the week since his story went mainstream. I believe he and I will be having his longest interview yet on this subject by a very wide margin. Maybe that's because he knows I'm not going to just ask for a few sound bites and then move on. And all this is happening as close to live as anything I've ever done. I'm recording these very words very late at night, right before posting EJ's interview. And tomorrow morning, I'm heading straight to Cambridge to sit down with Avi for 2.5 hours. I will post the main interview to the main podcast feed in a couple of weeks, which is my normal cycle. So Monday the 26th or thereabout, because there's a big holiday next week in the US and not a lot of people are going to be listening to podcasts. But I'm going to post a rougher version of the episode and probably a longer and more detailed version than the final episode that will go up on the main feed as soon as I can to the people who back this show on Patreon, maybe even as soon as this weekend. Because this is a very current story, and I'd like to get something out there to the most committed listeners to the show fast. So patrons, keep an eye on your feed over the next few days. Now, back to this great conversation we just heard with EJ Chichinilsky. As I said a few minutes back, I love EJ's work because not only is it flat out inspiring, but you can't help but learn a ton of neuroscience by exploring it. And that's a big reason why I do this show. All the research I get to do, it ranges from about 25 to maybe 40 hours per episode. That amounts to an amazing education, and I really learned an epic amount preparing for this episode. It was mostly self-directed, of course, but it was under EJ's very generous guidance. Now, this is the case with all of my guests, but EJ and I were in particularly heavy contact while I was bringing myself up to speed on his field. We first met when I interviewed him for this sprawling piece that I've been writing for the Sunday New York Times Magazine. And by the way, I'll tell you more about that article when it appears. It's largely complete, but its publication date is driven by editorial factors which are outside of my control and by news cycle factors which are outside of everybody's control. Anyway, EJ happened to pass through New York City while I was working on the article, so we were able to sit down in person. Then, not long after that, we both attended the same small neuroscience conference at Stanford. It was the very first instance of that conference, which is called the Brain Mind Summit. As a quick aside, I'll just say it was one of the most amazing events I've ever attended. It's an invitational thing, and if anybody listening to this ever gets an opportunity to attend the Brain Mind Summit, I urge you, indeed, I implore you to do so. The next one will be at MIT in May, and then the third one will be back at Stanford in the fall. I invited EJ to appear on the show after seeing his mind-blowing presentation at the conference. That presentation basically covered what we talked about in today's episode. So particularly those of you who just listened to the extended Patreon version, you probably know more about EJ's work than you would have learned at the summit, only without the PowerPoint slides. For those of you who are not backers of this show and listen to the shorter version, I'd of course very much appreciate it if you'd consider becoming a backer. And that doesn't have to be an act of charity, although I'd never say no to that if it enabled me to continue with this work, because $5 a month or more unlocks many, many hours of bonus content, which is only available to my supporters and which is hopefully worth every penny to them. Those recordings drop right into your smartphone, like any other podcast, which you subscribe to via a private link that Patreon sends you when you sign up. So it's really seamless, it's really portable, and it's really cool. If you sign up, you'll get access to the entire archive of bonus material going clear back to February. And I add to that archive every time I post a new episode. Sometimes it's an entirely freestanding mini episode, and increasingly often, it's an extended version of the interview from the main podcast feed, like today. I'm leaning into this extended version model more and more, because as the show's reputation is becoming more established, and as I'm gaining confidence as an interviewer, I'm starting to get a lot of time from my guests. Now, I don't want to overwhelm casual listeners of the show with super long episodes, but no way am I going to throw away any of the exquisite insights that my guests share with us, because their time is priceless. And by the way, so is yours. So the extended versions are absolutely worth the additional time for those who wish to access them. All of this is doubly important because for now, this show is free of advertising, as you hopefully noticed. So the only way I can support it is through backing at patreon.com, which for those of you who don't know the site, as you've probably inferred by now, it's a site that enables precisely this sort of voluntary support. It's a bit like Kickstarter, only for episodic content like podcasts. It is spelled patreon.com, and you can find my page at patreon.com/Rob Reid, and Reid is R-E-I-D. Thanks to my backers, this show's doing a bit better than break even. But due to the time requirements of each episode, I'm roughly clearing my hometown's minimum wage for the time I put into it. Now, New York City minimum wage is quite high compared to most communities, but it's not very high compared to New York City rent. So long term, this won't be sustainable unless I continue to gather more backers and or allow ads on the show. As I've explained in earlier episodes, the show's audience is big enough to attract some advertising, but I'm determined not to allow any advertising until I can really do it in a quality way. So as many of you have heard many times, I need to grow my audience. So if you're not financially able or just not inclined to back the show financially, you can still help it a lot by spreading the word about it. For each episode, I post a very self-contained tweet, which carefully describes the current show, and the tweet for EJ's episode is already up in my Twitter feed at twitter.com/Rob_Reid. So please consider helping this show light up new neurons out there by forwarding these little synopses around, and not just EJ's show. There are tweets for every episode I've ever done in my feed, and not a whole lot else, so it's pretty easy to find your favorite episode in there. Most of my episodes are only retweeted a few dozen times, actually, so I absolutely notice and deeply appreciate all relevant activity on Twitter. The last and most simple, easy, time-efficient, and free thing you can do to help this show expand its audience is to rate it on iTunes or on Apple's Podcasts app. Both of those apps frankly have rather squirrely UIs, but if you've made it through EJ's episode, or frankly any of my show's episodes, you are definitely smart enough to figure it out, I promise. These ratings matter to Apple's promotional algorithm, and therefore they really do help the show expand. So that's it for now. I hope you'll join me next time.